Good morning. Welcome to my class. Let's talk about scientific method. Several versions out there. None of them really set in stone. All of them follow kind of the basics. The reason you're doing this is to go through an orderly method to obtain information that's verifiable, explainable, one that could be accepted. These basic steps include the observation period and understanding that we have cause and effect. When something's present, we'll get a different effect. Two groups, something's different, we get a different effect. Everything else is the same. Little change, different effect. We can link it back to each other. Second thing is to propose the problem. After observing, you want in some way to bring out, through a little bit of brainstorming, what the problem is, what you're observing, and then you'll move right on to formulating a guess, an educated guess. You would look at what is already out there so that your problem and your guess at the solution will be backed up by previous knowledge that's already been done. Don't reinvent the wheel. That's spinning your wheels. That's being in a rut. Let's move forward. Let's push the envelope a little bit. After observing and stating your problem, you will come up with a possible solution. And we're going to call this possible solution right there, hypothesis. See how it's spelled H-Y-P-O, hypo, under thesis. So it's kind of like an understudy. And once you have your hypothesis up, you need to design an experiment. This experiment needs to test your hypothesis. Set up a couple of groups, change a little variable, as they're called. Best to only change one variable. That way you can kind of pitch it as a yes or a no, a straight cause and effect. That's going to take some brainstorming. After you run your experiment, you then will get your data, and your data will either tell you you're on the right track or that you need to go back and you need to revisit your educated guess. Even failure at this point is important, and I say failure, it might be unexpected results, will be important to you in your search for that verifiable knowledge. It'll move you closer. Time to come to a conclusion. Using the data that you've got and your interpretation, your analysis, you'll move towards your answer. Now, your answer may force you to adjust your hypothesis according to what you already did, and guess what? You loop back to altering the hypothesis, which will then give you a different experiment to sort of isolate the cause and the effect change. Once you have gone through this and you feel as though you can back up your hypothesis, the idea is to publish your results for peer review. Your peers are folks that are in the field, or, you know, for most of us that would be telling our friends, or that would be letting folks know. But in the biological world, you would put it in a journal, and you would have it in front of everybody. You would keep it real clear and clean and allow them to go ahead and to test your hypothesis and your educated guess and look at your work, and they may come up with a, hey, 2 thumbs up. They may come up with a, not so quick there. I see some flaws. So we're at that point now where other people are testing it, and if others validate it, you can move on to a theory. Ooh, that's a good thing, and you want to do that, you know, and as you're doing that, you may get doctor in front of your name, PhD, and those kind of things. No doubt maybe your salary increases if that's the kind of job that you have. And after it becomes widely accepted, you then can move into what is known as a scientific law. Woo! And perhaps you could name it after yourself. You could give it your name, and then it's in perpetuity for your grandchildren to see and say, well, that was my grandpa that did that. What fun. A quick picture of a scientific flow chart, the problems start as you expect. Choose your topic, identify your problem, research the problem, develop your hypothesis, then design your experiment, test the hypothesis, analyze your results, and formulate your conclusions. As you can see, it does loop back in case you need to adjust. Thank you, makeitsolar.com, for your beautiful drawing. An important part of the study is to look at the experimental design. Typically, you will have two groups. One group will be called the control group, the other will be called the experimental group. In both groups, you keep like conditions. They are the same. The like conditions will be called the dependent variables. They don't change from one group to the other. You will, however, have the one change. You'll change some factor, and that some factor is known as the independent variable. So the outcomes over here versus the outcomes over here, between the two groups, there will be a change. And what you want to do is the change in the outcomes, you then can link them to the independent variable because of cause and effect. It's important, just one variable. Isolate one so that you feel confident that that one variable is the reason for the change in the outcomes. Let's move on to a simple experiment just to illustrate what we're talking about here. I've got myself a problem stated. What is the effect of fertilizer on plant growth? And then I come up with my hypothesis. And I say, hey, fertilizer will increase growth of plants. Good. I'll set up two groups. I said two. One group will be the control group. One group will be the experimental group. I have to keep everything the same in my dependent variables, and things like soil and light and plant type, temperature, pH, water, and atmosphere. Those kind of things you don't normally think about, but don't change them between the two groups. Keep them the same. That's the idea. Now, the one little change I will call the independent variable will be my fertilizer. Sounds good. So I'll end up with a cause and effect as I run through my experiment. I'll collect my data, my observations. I'll either support or I'll discredit my hypothesis. I may come up with some new questions. I may have to rerun the experiment just a little bit different. I mean, in fertilizer, I don't know if you know this, NPK, nitrogen, phosphorus, and potassium are the big macro, I'm using the word macro, which means large, macronutrients, and perhaps after running this experiment, I can take a look at varying the NPK of the different fertilizers, and that may lead me into a whole other line of research. Perhaps I can get a grant for it and with an expense account and go to a symposium out in Hawaii and then take my show to Japan, and won't that be fun?